But no, thanks so much for coming along on our Friday afternoon, and a massive thank you as well to Lucy for being such an excellent host. She is probably the most organised person I've ever met in my entire life. Like, I'm talking Gantt charts here, people, Gantt charts. So thanks so much for giving up your Friday afternoon to come along and hearing all about microorganisms with me today. So I'm just going to be covering a complete basics for people who have never sort of looked at this before. And then we're handing over some case studies both on the water side of things and the sewage side of things. And this event today is both with the Institute of Water and the Royal Society of Biology. So there might be a lot of people in the audience who are maybe new to the water sector or maybe new to biology in general. So I'm just going to kick off by sharing some slides. So please excuse me while I do a little bit of switching my screens around. There we go. You should be able to see that okay. I'm also like not used to Zoom. I've been using Teams so much at the moment. So here we are, water underneath the microscope. Who am I? Why am I here talking to you today? Normally I would just sort of plow on into my presentation, but I thought it was important to be like, I kind of know what I'm talking about. So this is kind of what I've done. I've been working quite a bit in microbiology, and the reason why I was really passionate about putting on this event today is because I really enjoy sharing my interests with other people, and microorganisms is something I'm super excited by. So normally if I'm going into schools and delivering something similar like a basic introduction to microbiology, I generally take about 6 hours, a 6-hour-long course sort of doing this. And obviously today I've got 20 to 25 minutes. So it was really difficult for me. It's almost like talking to a Star Wars fan or like a Game of Thrones fan and like getting them to explain the key concept behind whatever franchise it is they're excited by. It's very similar like that for me. But yes, I've got this background in microorganisms. I'm currently working with a company called QCL, and I work with a whole suite of different microorganisms in the water industry. And yeah, I'm coming from like an academic background, but also with a heavy dose of industry in there as well from Anglian Water. So yeah, let's kick off today. So I was trying to have a think, and a bit like the whole fandom thing, I was sort of like, I don't really know where to start today because I've got so much content and so much stuff that I would like to tell you guys about and to teach you all about and for you to understand. But I had to have some kind of a starting point. So in the chat, I just want you to write if you understand this or not, or if you don't want to use the chat, just have a think about it yourself and just think, do you understand this? So I literally just typed in E. coli, one of the most commonly used microorganisms, especially in the water industry sort of sector, into Google, and it came up with this paragraph, and that paragraph should be the basic description of what this is. It's from Wikipedia, the best source of all information, as everybody knows. And I was like, actually, for people who are new to microorganisms, this isn't a good description at all. This is actually really confusing. So I thought my goal for today's session, rather than trying to teach you everything there is to know in the world, what I want you to be able to do at the end of my presentation today is understand what this little paragraph is actually saying. And along the way, I'm going to give you some tips on how to think like a microbiologist, or how to pretend like you think like a microbiologist. Just some of the practices, just some of the ways of thinking that you do if you're into microbiology. So that's my goal for you guys today, is to understand this little paragraph here. So the first little bit is this abbreviation thing. So the actual naming of microorganisms. As someone who's done like quite a bit of teaching during my PhD, the naming of things was super important for me, and it's something that biologists in general, I feel, can be a little bit, particular about. So all organisms have these two names, the genus and the species. So we've got E. coli there as an example, and the font choice is also really important. So you'll see there that the genus is capitalized, whereas the species is lowercase, and the italics there. So here's a good example of those different things. So this is Homo sapiens, humans, and these are the different kingdoms and the different classifications of those organisms. And this is basically how all organisms that exist are named. So a bit of general biology rather than microbiology. So you can see there that we're eukaryotes, we're animals, so we're in the animal kingdom. Chordata, phylums, that basically means that we've got like backbones, vertebrae. We're mammals, we're primates, and so on, and that's how it's named. And microorganisms, it's exactly the same. So they have all these different different groups of all these species, and then you narrow down and narrow down and narrow down until you get to like a species level, and that's why they're named like that. But we quite often abbreviate, so we just have E. coli as an example. So moving on to the next little bit of our little paragraph that we've got is gram-negative. What does gram-negative mean? So in biology, we're super keen on classifying things. I mean, how are you going to get you know, the next microorganism named after you unless you study taxonomy? So we're really all about categorizing, classifying, naming different species, differentiating between the species, misnaming species, thinking different things are actually the same, and then later finding out that they're not. That's something that it reoccurs and reoccurs. So the gram-negative part of that sentence. So the gram reaction is basically just applying a dye, a bit like my purple hair dye that I've got on at the moment, and seeing whether those microorganisms uptake that dye or whether they kind of like reject that dye and stay the colour that they already are. So this is just one way to identify between different types of bacteria. And it's basically just due to the differences in the cell wall composition. So what are the actual components of the cell wall? What is the thickness of that cell wall as to whether it can uptake that dye or not uptake that dye? But this is just one way of categorizing specifically bacteria. And like I said, more on that in just 1 second. So one thing that's really important to consider if you're sort of working in the field of microorganisms, or maybe you speak to microbiologists in your role, is that there's a lot more out there than just bacteria. So we quite often focus on bacteria because, well, for a start, it was the one that we knew most about. So we did a lot more research into them, we really understood them a lot better. Obviously, at the moment, there's a sudden like uptake on the interest in viruses, which is relatively new for sort of the mass population, I would say. But quite often in the past, when you're speaking to people about microorganisms, they just kind of assume bacteria, and that's why some other species are so vastly understudied. So some of the researchers that I was working with during my PhD were looking at things like fungi, which are super interesting. But we don't really know all that much about them. And there's loads and loads of them out there. These microorganisms literally thrive everywhere. But even when you're looking at things like adverts for, you know, cleaners that are killing microorganisms, they're just talking about bacteria. And they quite often use the two terms interchangeably. But actually, there's loads of different types of organisms out there that don't come under that bacteria sort of bracket. So, for instance, we can categorize these different microorganisms in different ways. So for viruses, we look at their genome, specifically if it's single-stranded or double-stranded. Whereas in fungi, we're looking to see if it's one cell carrying out all the functions or many cells. So an example of many cells carrying out a function would be the humans. I'm not saying humans are fungi, I'm just giving an example of many cells you know, that an organism might have. So we've got like white blood cells, you know, skin cells, fat cells, etc. So we're made up of lots and lots of different cells. Whereas, you know, a yeast, as an example, each individual yeast is equally doing the same role as every other little individual yeast. So as you can see, you know, that gram-negative, that gram-positive is just one way to categorize bacteria. But actually, all these different microorganisms that exist also have all their own different classifications of things as well. So that's that next bit. So this next bit is all about anaerobic bacteria, and this might be something that we brush on a little bit later in Alex's presentation, all about sort of the wastewater environment. So aerobic bacteria need oxygen to be able to survive. Whereas anaerobic bacteria do not, they're able to do it without the presence of oxygen. I'm just checking the time to make sure I'm running okay. Yes. Whereas, so this E. coli that we're talking about today is just this example, can actually do both if it needs to. And I think that's a really good example of how adaptive microorganisms are. So microorganisms require different food sources So that could be things like carbon. Carbon is like the key one that they need. Carbon is one of the building blocks required for life. But there's also other things as well, like nitrogen, phosphorus, sulfur. There's also like trace elements as well. So you'll get specific organisms that are adapted to living on iron. I'm thinking like cast iron pipes here. And there's all sorts of microorganisms that live in really specific, really weird, really niche environments. So one you've probably heard of is hydrothermal vents under the ocean, on the ocean, like the seabed, really, really deep, really, really hot environments. So there are different microorganisms that are adapted for really high temperatures, really low temperatures, really high pH, really low pH, and so on. So a bit like, I don't know if you've heard of this term because I'm not sure if it's normal knowledge or not, but if you've heard of the term Darwin's finches, so Darwin sort of identified that these finches had different beak shapes for different food sources and for gathering those food sources. So for instance, I don't know, something that wants to get at insects within wood might have like a really sharp, really pointy beak so that it can get in the holes to eat the wood. So it can get in the holes to eat insects through that wood. Whereas something that's, I don't know, eating fruit could probably have a shorter beak. So these, although being the same species, they've adapted different traits to be able to feed on slightly different food sources. And microorganisms are the same in that they've all adapted to live in these different environments and have these different niches, is a word that we use quite often in biology and in microorganisms, different niches, different environments, different food sources, different places to live. So everything really is in balance. So one key tip that I've got for you today is to always think about microorganisms. This is something that I personally do. You should think of it like a food web. So if you're ever trying to think about how a microorganism is adapting to a specific environment, or you're not really sure what their behavior is doing, instead of thinking of it as this, so a microorganism is something that's so small that you need to look at it underneath a microscope. Rather than thinking of it in terms of that, think of it in terms of things that you do understand and you do know the behaviors about. So one example is a plant. You know that a plant needs water, soil, a food source, it needs the sun, and so on. So when I was doing my PhD, I had these fake pipe environments, and I was explaining to the operators on site that if the facilities went offline, all the biology was going to die within my pipes and it was going to impact my experiments. And the reason for this is because it's the same as the plant analogy. If you stop watering your plant, the plant's going to die. So if you just relate it back to things that you do understand, then that really helps a lot when you're trying to understand its behavior. So this is a basic food web of just the different environment. You can see here there's lots of plants being eaten by squirrels, by grasshoppers, and then the squirrels and grasshoppers then being eaten, and it goes all around in circles. But if you have some sort of imbalance here, different species are going to pop up. So for instance, if something happened to the squirrel population and they really, really dropped off, that's going to impact their eagles. They're going to have and the foxes, they're going to have a reduced food source there. And equally, it might impact the plants because they're not having as much, not having as many squirrels eating them, which could then cause a boom in plants. So all of these knock-on effects are happening because all of these different species are in balance. And it's the same with microorganisms, especially within like a contained system. So within a pipe network, as an example, or equally, in the air around you, or on a moldy sandwich. So there are all sorts of different microorganisms in all of these different environments. And if one of the species changes, another species can come in place. So for instance, say if you have a microorganism that eats oxygen and gives off sulfur, you might have another microorganism that eats sulfur and gives off oxygen. And then those two organisms are symbiotic. Just as there are many examples of animals that are symbiotic, that work together to achieve the same outcome. So if you just think of it in terms of that, that's really helpful for understanding what's going on. So another water-based example might be you've got some microorganisms who are living on a cast iron pipe, and some of them are using iron as a food source, which is then making the iron more available for other microorganisms who are only able to survive because of that iron. But equally, you've got microorganisms eating, predating on other microorganisms too. So it is very much like a food web. And I'm not going to speak to you about all the different organisms out there and what they're doing and how they're eating. But if you're interested, get in touch, because we could talk more on this. I could talk a lot on this. But yes, that's how you should think about things. So yeah, there's only so much food available. There's only so many niches, so many like places that things can live. Everything is in competition to survive. At the end of the day, everything just wants to survive. And the environmental conditions, what I was talking about earlier, that temperature, that pH, those sorts of things really do matter and can vastly change the speciation, the different species of microorganisms present in an environment, favouring one, selecting for the decline of another, just as an example. But yeah, they really matter as well. So that was a very long one. I hope you're still with me. I know it's a Friday afternoon. Apparently the weather was going to be nice today, but it's not so great here. But I hope you're still awake and with me. So next one is rod-shaped. So unlike some of those other words, which are really quite intense and there's a lot for me to go through, that one is literally just the shape of the organism. So they can come in lots of different shapes. Here's a great example of some of the different ones. I've got some microorganisms with me today because I'm a big nerd. So these are some microorganisms that I've got. And as you can see, they're not this big, they're blown up to size, and they're not this cute either. Like, look at the eyes and belly. Yeah, on that Ebola, he's nice. So you can see they're all like different shapes, different sizes. They've got different things going on. Some of them are motile, so they've got flagella, so they like can squiggle around. Some of them don't. But yes, they're all really different from each other. They come in lots of different shapes and sizes, but all of them small as a size. So that's what rod-shaped is. It literally just means shaped like a stick. Next one is coliform. So coliform bacteria, their sort of definition, is a type of gram-negative bacteria that can be frequently found in the feces of warm-blooded animals because they're known to inhabit the GI tract. Coliforms don't normally cause serious illness. I mean, there are exceptions, for instance, immunocompromised individuals. So it could be like the really young, the really sick, the really old, just as an example. But they are really easy to culture. So coliform bacteria is a frequently used indicator organism within the water sector. And the reason for that is because we know they've got this link with poo. So we quite often say that if there is that coliform bacteria there, then there's a possibility that sample has been in contact with poo at some point. So we use it as an indicator species within the UK. So what does that actually mean? So you can see there in the image, what you do is you get, and this is the way that we've been doing this for many years, you get a plate and you get an agar jelly. So that's basically, it is a jelly with all the nutrients needed for a specific microorganism to live on and grow. So you'll get your agar, you'll spread over a water sample onto it. Maybe it's been taken from a customer tap or from a tumour works or anywhere. I remember in uni, we did it from our fingers. We like imprinted our fingers on the agar plate to see what disgusting things people had been growing on their hands and really like call out the people who hadn't been washing their hands when going to the toilet. So you take whatever imprint or whatever sample you want, you put it on the agar plate and you leave it in a nice warm place to grow. So like I was talking before about these different microorganisms requiring different foods, different environmental conditions, you're giving these plates the ideal conditions that specific organism needs to grow. And then you're just letting it grow and reproduce. And then you're counting the number of colonies, like the little groups of cells that are then forming on that plate. And that's how we sort of work out with this plate counting technique if there's coliforms, if the water is safe or not. Now, this is what we do in the UK, but there are lots of different indicator organisms that lots of different other industries use, and also different countries use different things as well. So in the Netherlands, for instance, they don't use E. coli as an indicator generally. Another one that we can use quite often is like enterococci is quite common for us. But yeah, those are just some of the different microorganisms that we're using. And the key reason is they're just easy to culture. We've been using this for many, many years. And there are different techniques out there that we're developing at the moment that sort of go above and beyond that to get, I don't know, something that happens like a microbial count a little bit quicker than this whole waiting for the culture to grow up. And there have been lots of research as well on just because E. coli is present in the sample doesn't mean, it doesn't mean, well, for a start, that it's harmful, but there could also be a sample with something quite nasty in there that doesn't have E. coli there. So it's a good indicator organism. We've been using it for many, many years, but there are loads of different new sort of techniques that are springing up with lots of exciting research that people are doing to see if there is something that works just as well, if not better, if not quicker. So yes, this is sort of why we're focusing on coliforms generally in the water industry, and so why we're focusing on them and what that focus kind of looks like for us. And I think Nicola will be touching on this some more in her presentation as well. So my question for you all really is, do you now understand this paragraph of text? Because we've gone through sort of line by line to talk about some of these things, what they mean. So yes, I'd like you to maybe revisit what you wrote in the chat at the beginning, or what you wrote down in your notebook, or what you're thinking about, to then think, do you understand this now? And if you don't, maybe put in the chat why you don't understand it, or maybe what you've not particularly got your head around maybe. So I think some final tips from me, this is what I thought was really important before sort of moving on to some of the more exciting presentations that we've got today from our specialist experts, is when you see a paragraph of text like this, don't panic. It's a lot of long words. I mean, it's a lot of Latin. And for people like me, you know, I never studied Latin at any point. I don't know any Latin whatsoever. And they're long words that you don't particularly know that are quite scary. So for a start, don't panic. Most people don't know what they mean anyway. That's why, we abbreviate everything anyway. So many abbreviations. But I think one of the most important things is to think of microorganisms like animals and plants that you do understand. At the end of the day, they need food, they need somewhere to live. They're going to compete. They're very stubborn. They will survive everywhere, anywhere. You'll look at a place where you're like, no, nothing could survive this. I'm pretty sure nothing could be living here, and they will be living there. So they are everywhere. One of the key ones, well, for a start, getting that nomenclature right, so making sure that you're doing like the italics and the capital letters and the lowercase thing, because it's super pedantic, but that's one way that you can spot someone who doesn't necessarily know microorganisms when they do that. So that's a sneaky tip for you from me. And there's a lot more microorganisms than just bacteria. That is kind of like a bit of a bugbear really, like don't use the two as the same word, use them as separate things. And yeah, look into some of that exciting research that's going on in some of those different microorganisms, because it is really cool. And finally, and we will cover this in some of our presentations today, they are not all bad. They can be really helpful. I mean, if we didn't have microorganisms, we wouldn't have, I don't know, we wouldn't have bread, beer. We'd have no beer, guys. They're really important. So yes, they're not all bad. But I think our two presenters that we've got today will do a really good job of showing sort of like the negative side of microorganisms as well as the positive side of microorganisms. So I hope that's been okay. Like I said, normally I would have given you like a very long 6 hours all about microorganisms and what they do and who they are and how they live their lives. But instead I've tried to distill to you what I think are the key things, almost like my top tips for speaking to a microbiologist. So I hope you've enjoyed today's presentation. Do feel free to get in contact with me, email me, tweet me, message me on LinkedIn. But no, thanks so much for coming along today.